Today we're diving into one of the most complex battlefields you can possibly imagine, the war on cancer. And it's a war fought not by outside armies, but by the cells inside our very own bodies.
You know, we always think of our immune system as this ultimate guardian, right? Our protector. But what happens when that system gets corrupted? That's the huge question we're tackling today. Can our body's best defenders actually flip sides and become cancer's greatest allies?
So, to really get a handle on this, we need to shift our thinking. Let's stop seeing cancer as just some foreign invasion and start seeing it for what it really is, a kind of civil war. A brutal conflict where the loyalties of our own cells are constantly up for grabs. Day in and day out, your immune system is doing something absolutely critical called tumor immunosurveillance. I mean, think of it like a 24/7 security patrol inside your body, constantly identifying and just zapping any rogue cells before they have a chance to become a real threat. It's our first line of defense, and honestly, most of the time, it works beautifully.
But, and this is the big but, here's the twist. Sometimes this system doesn't just fail. It breaks down in a way that is just shocking. It's not that the immune cells simply miss a cancer cell. Oh no, it's that they are actively recruited by the tumor, corrupted, and then they start to help it grow, help it spread, and help it survive. They literally switch sides.
Okay, so you're probably asking, how on earth does a loyal soldier turn into a traitor? Well, that switch gets flipped on a very specific, very chaotic battlefield. It's a place known as the tumor microenvironment.
The tumor microenvironment, or TME for short, isn't just the tumor itself. You have to think bigger. It's this whole complex ecosystem all around it. You've got the cancer cells, sure, but also blood vessels, structural tissues, and a whole army of different immune cells, all packed together, all interacting. This is the stage where the entire drama plays out.
And the key players, the boots on the ground arriving on this scene, are the tumor-infiltrating immune cells. Let's just call them TIICs. These are the immune cells that actually leave your bloodstream and march right into the heart of the battle, embedding themselves right inside that tumor microenvironment.
And this, right here, is where it gets fascinating. Because once those TIICs are inside the TME, their fate is up in the air. They can become heroes, or they can become villains. So how do we unmask these double agents?
Just take a look at this. This perfectly shows the two faces of one key cell, the macrophage. Over on the left, you've got the M1-like macrophages. These are the good guys. They fire up the immune system and lead the charge to kill cancer cells. But now, look to the right. The tumor can actually corrupt these exact same cells and turn them into M2-like macrophages. These are the traitors. The tumor just bombards them with signals, reprogramming them to shut down the immune response and even help the tumor build new blood vessels to grow and spread. It's the same cell, but with two totally opposite jobs.
Now, the hero team is led by cells like cytotoxic T cells, or CTLs. Think of these as the immune system's special forces. They are trained to seek and destroy cancer cells directly. And they get help from T helper cells, who act like the field commanders, calling the shots and coordinating the whole attack.
But the villain team, well, they have their own elite units too. The most notorious are probably the regulatory T cells, or Tregs. Their whole job is to pump the brakes on immune responses. Now, in a healthy body, that's great. It stops you from attacking yourself. But inside the TME, the tumor hijacks them to call off the attack, basically creating a protective force field around the cancer.
So when you look at a complex diagram like this, it really boils down to one simple, critical idea. On one side, you have cells like those treacherous Tregs in M2 macrophages. Their presence means a worse outcome for the patient. But on the other side, you've got the heroes, like cytotoxic T cells and M1 macrophages, and they point to a much better outcome. The balance between these two armies can literally decide a patient's fate.
And the really crucial thing to get here is that this isn't random. It's all orchestrated. There's this incredibly complex network of chemical signals, some that say attack and some that say stand down, all happening at once. The tumor itself is a master manipulator, sending out signals to corrupt those incoming immune cells, turning would-be attackers into loyal protectors.
Okay, so if understanding this battlefield is the key to winning the war, that brings up the next big question. How in the world do scientists figure out who's on which side? How do they actually count the soldiers?
Well, the old school way was painstaking, to say the least. Scientists would have to stain these tiny tissue samples and then physically sit at a microscope and count 1, 2, 3 different types of cells. It was incredibly slow, it was subjective, and you could only look at a couple of cell types at once. It gave you a really incomplete picture. But today, we are in a completely different world. Thanks to these incredible leaps in gene sequencing, scientists can take a tumor sample, analyze all of its genetic activity, and then use these powerful computer tools to create a detailed census of every single immune cell type that's there. It's like creating a unique immune fingerprint for every tumor.
So, the process is basically a four-step mission. First, you gather these huge amounts of genetic data from tumors. Second, you unleash powerful algorithms to sift through all that data and get an accurate count of every type of immune cell. Third, you use that count to build a predictive model. The question is, can this fingerprint predict survival? And then fourth, and this is the most important step, you test that model against real-world patient samples to make sure it actually works.
And here's a little peek at what that looks like in a real research lab. You've got scientists pulling data from these massive public databases and feeding it into super sophisticated tools. These tools are the census takers, counting up all the different immune cells. The output is what allows researchers to connect a specific immune fingerprint to a patient's actual chances of survival. It's incredible!
But get this. Just when you think you've got it figured out, there's another layer of complexity. An immune cell's role isn't even the same across different cancers. Look at this biomarker HMGB1. If you have a lot of it in bladder or prostate cancer, that's linked to an unfavorable prognosis. Not good. But in gastric cancer, having a lot of that same exact biomarker is linked to a favorable prognosis. It shows that context, the type of cancer, changes absolutely everything.
So, what is the endgame here? What is the point of all this insanely complex research? Well, it all comes down to one thing: moving beyond one-size-fits-all treatments and creating a truly personalized battle plan for every single patient. See, this immune fingerprint isn't just some academic curiosity. It is an incredibly powerful biomarker, which is really just a biological clue. And by reading this clue, doctors can start to predict how aggressive a cancer might be and, crucially, who is most likely to respond to game-changing treatments like immunotherapy.
And this is all ushering in a brand new era of cancer care. We're talking about predicting patient outcomes with much better accuracy. We're talking about matching the right patients to the right immunotherapy, so you don't waste precious time on a treatment that won't work. I mean, just imagine two patients with the exact same diagnosis of lung cancer. In the past, they might have gotten identical treatments, but now, a doctor can read their immune fingerprints and see, patient A's tumor is full of hero T cells. They're a perfect fit for this immunotherapy. While patient B's tumor is swarming with traitor macrophages, meaning a totally different therapy would be a much smarter first move. That's a total game changer.
We're learning that winning the Civil War isn't just about fighting the enemy anymore. It's about turning the enemy's own allies against it. This knowledge is completely transforming how we fight cancer, and it leaves us with this one truly inspiring thought. If we can read the battlefield and actually reprogram the soldiers, what will we cure next?